Welcome to this review week for the first lab practical exam. I hope as you look back over the last 6 weeks of labs that you are proud of yourself, that you appreciate how much you have learned, how hard you have worked to learn all that has been asked of you as we study the human body. One way that I oftentimes think about anatomy and all that we're learning is to think about all of the pieces, all of the lists, all of the facts that you've been asked to learn. It's like opening up a 1,000-piece puzzle. At first it seems overwhelming and you wonder how you'll ever put this thing together. We have a guide, but the idea of putting it all together is overwhelming. But we've all put together puzzles. We know that one of the first things you do is find those edge pieces. You look for those pieces that have straight edges on them. You then start looking for corner pieces and you start to outline all that you need to learn. And that's what you've been doing all semester. You have been marking and highlighting and learning about all the things that we've been asking you to look at. You've been looking in your lab manual, in your lab atlas, looking online, gathering information, and slowly, the puzzle has been coming into focus. Now, I know that if I put a large puzzle on my dining room table, that if I don't sit down every day or two and put together a small part of it, that the puzzle will not ever come together. I need to sit down and focus on putting together a small section of that puzzle. Maybe I'm putting together like colored pieces or pieces with similar designs. We've all done this. And over time, as you put together more and more of those small sections, the big image begins to come into focus. And eventually, we put it all together. And that's where we are. In this time of the course, we're now asking you to put all of this together. The lab exam is going to be a comprehensive exam that's going to cover everything we've done from labs 1 through 5. So let me quickly go through just a list of these main ideas and topics. You have this same document in your lab review folder. It's called the checklist. So let me just walk you through this checklist and you can begin to gather all of these components. I would ask that when you come into this lab review week that you have perhaps put little post-it notes on the key pages that have these main ideas on them in your lab manual, in your atlas, anywhere where you have been keeping notes. It's important that you are now pulling together, if you haven't already done this, pull together all of your materials for studying. So let's go back to Lab 1. Lab 1, we figured out how to get into Zoom. We got to meet each other. We then went through the introductory materials. This material focused on basically anatomical terms. We learned about directional terms. We learned about the body cavities. We learned about those 3 serous membranes and the 2 layers that make those up. We learned about the planes of section, the directional terms, anterior, posterior, ventral, dorsal. Those were also important components of the first exam. So that should be material that you have learned well. Also in the first lab, while it wasn't on the first exam, you were also expected to learn the parts of the microscope and what those different parts do. Finally, there was a simple calculation of how total magnification is calculated. You may recall that total magnification is the ocular times the objective lens. In Lab 2, That was the lab that was completely on mastering and involved your completing a number of worksheets, watching a number of videos. This lab dealt with a lot of the basic foundational ideas within biology that apply to anatomy. And in this lab, you were asked to know about the cell cycle. You were asked to know the stages of mitosis. You were asked to learn about osmosis and diffusion. And you were also asked at the bottom about the pH scale. You were asked about solubility, about hydrophobic versus hydrophilic, and water-soluble versus lipid-soluble molecules. Those topics that I just mentioned were also a part of exam 2. So again, these are topics that you should feel comfortable with. You have studied these not only for your lab quizzes, but you've also seen these topics in your lecture exams. Lab 3 was the histology lab. Recall that you were asked to identify 7 different epithelial tissues and know the locations and special characteristics of those tissues. You were asked to know and identify 9 connective tissues, also their locations and special characteristics. The 3 types of muscle, their location, their modalities, that is whether they are voluntary or involuntary, and then again, special characteristics, think striations or intercalated discs. You were also asked to simply recognize nervous tissue. You also looked at skin very briefly. You were asked to compare scalp, that is thin skin with thick skin. And finally, this is the lab where you had worked through the field of view worksheet. Remember, all the answers are provided for you, and you need to be able to approximate the size of an object under the microscope when you're using the 4x, the 10x, and the 40x objective lenses. So that was Lab 3. Lab 3 was also a very large part of Exam 2, which you have finished up in the last week or so. So again, hopefully histology is something that you have become familiar with and very comfortable with. Moving on to the more recent materials, Lab 4 dealt with the skeletal system. In Lab 4, you learned the names and the locations of 200 of the 206 bones. You learned about vertebra. You learned how to distinguish cervical from thoracic from lumbar vertebra. You learned the special characteristics like the dens, like the lamina, the pedicle, the spinous process, the transverse process, all of that list of terms. And you were asked to take an extra special look at the structures of bone under the microscope, so you know a little bit more about compact bone histology like lamella, osteon, canaliculi. But you were also asked to know about long bone anatomy. That is, what are the macroscopic structures of bone? Think the epiphysis, the diaphysis, the nutrient foramen, the bone marrow, things like that. So that is Lab 4. Lab 4 is also an important body of knowledge related to Exam 3 in lecture. And then lastly, the labs that you've been dealing with the last couple of weeks. Lab 5, broken up into 2 parts, Lab 5A and Lab 5B. Looking at this list in total from everything that you've done in both labs, you should be able to identify the 3 types of muscle tissue. Again, reviewing the muscle histology, being able to recognize skeletal versus cardiac versus smooth muscle under the microscope. You should know the different movements that the bones and muscles allow us to do through our joints, and know the 6 different types of synovial joints and examples of each. you've been working hard on learning a long list of bone markings that are on both the appendicular and the axial skeleton, and you have been working hard to identify muscles. Of those muscles, there were 20 total that you were asked to know their actions. So specifically know the actions of just the 20 muscles that were specifically listed, as well as know the specific origin and insertion sites for the 9 muscles that were indicated between the two lists. Finally, you were given a number of structures to learn from 4 major joints: the knee, shoulder, hip, and elbow. Wow, that's a lot of material. So how will this exam be structured? This exam will be like a lab practical exam. That is, an exam where everybody comes into the room, walks around the room, moves from station to station, and must answer the questions as they move around. There's no going back. Well, to replicate that experience, the exam will be similar. There will be 102 questions. It'll count out of 100 points, so each question is worth 1 point. You could earn a couple of bonus points. Different from the lecture exams where you have the entire exam open to you and you can scroll up and down through the exam, the lab exam will be a forced completion format. That means that you must answer a question in order to move on to the next question. So you won't be able to go back. You'll have to answer the questions as they're presented and move forward. 80 to 85% of the exam is derived from labs 3 through 5. That is histology, bones, bone markings, muscles, origins, insertions. That large body of knowledge will make up 80 to 85% of the exam. So that means, yes, there'll be a question on pH. There'll be a question on mitosis, a question on cell cycle, a question on directional terms. But the ideas that were presented in the first 2 labs will not be equally weighed on this exam. The majority of this exam will be labs 3, 4, and 5. The images that I will use for this exam are taken from ones you have already seen or should be studying. That is, there are images in your lab manual, lab atlas, PAL on mastering, the Martini textbook, the lab presentation PowerPoints, or any of the lab files that have been given to you. All of that is where I will grab the images and ask you to identify structures. Go back and review all of the content folders in the labs. Go back and look at your pre-labs that you submitted. Go back and look at all of your lab manual work that you did in or outside of lab. And then finally, before you take the exam, make sure that you complete the PAL quiz on mastering. Just like you have quizzes to do on mastering before each lecture exam, there is a quiz, or I really should just call it an assignment, that you'll complete on mastering. That assignment is open book, untimed, and that means you can come and go to it, do a little bit at a time, and break it down. It's meant to be one more round of review before the exam. The lab practical exam will require Respondus Lockdown Browser. You will find it in the lab practical folder on mastering under lab materials, and it will open up at Friday noon and go through Sunday midnight. Now, this is the same weekend that you'll also be taking lecture exam 3. And yes, I know that they're both on the same weekend. These 2 exams, the great thing about them is that the vast majority of these exams are both over the muscle and skeletal systems. And so you are studying for one while you study for the other one. So as you think about that puzzle and you're putting it piece by piece and bringing it all together, hopefully before you take the exam, you are sort of looking at this image and saying, okay, I've now got this put together. I understand the connections, the interrelated connections between histology and the skeletal system and the bone markings and the muscles. And I understand how the origins and insertions are the attachment places for the muscles to attach to the skeleton to do the actions that have been described. I hope this little video is giving you a plan, giving you some ideas on how to prepare. And in the lab this week, you'll be doing a number of activities that will also help you reach your goals. Also, make sure you get your pre-lab turned in as is described in the lab folder. Do very well on this exam.